Hello everyone, welcome to this wonderful session, Part-3, Chapter-2, Biological Classification of Unit 1, 11th Standard. In this part, we have to discuss heterotrophic mode of nutrition, reproduction in bacteria, economic importance of bacteria. Then we have to see blue-green algae, that is cyanobacteria, and then we have to see mycoplasma. You know, children, already we have discussed that is autotrophic nutrition in bacteria in part 2 of this chapter, and of part 1 we have discussed that is classification systems. This chapter, as usual, will be completed in 7 parts. After completion of all 7 parts, we have to discuss questions of NCERT exemplars, and then we have to see previous year examinations question. Chapter 2 of Unit 1. Chapter 2 is biological classification. Of this chapter, Part 2 we have discussed in the last class, where we have discussed that is autotrophic nutrition in bacteria. Already we have discussed that is autotrophic nutrition in bacteria, and there you have seen that is photoautotrophs and chemoautotrophs. We just want to justify that bacteria, these are very simple in structure but metabolically diverse these are. So nutrition wise you have seen that different type of nutritions are there. Now in continuation of that we have to go for this part 3, that is heterotrophic nutrition in bacteria. Heterotroph, they are not able to make their food. Heterotrophic bacteria are most abundant in nature. Important question, that is most abundant one. Majority of these they decomposes, that is complex substances are simplified, and the main categories of these that is bacteria are that is saprophytic, symbiotic and parasite. Saprophytic one, you know the term, these are the free living bacteria. They obtain nourishment from organic remains such as dead animals and plants. They will be that is simplified, they will be digested. They are responsible for mineral cycling. These are responsible for that is mineral cycling. So among, you see, the heterotrophs are most abundant, and among heterotroph, the most abundant one, these are saprophytic one. Symbiotic one, the two organisms living together, the bacteria that used to live with some that is other organism, and this other organism that is used to provide something to bacteria, and in turn the bacteria used to provide. This association, you know, is known as symbiotic association. A very popular example is Rhizobium leguminosarum. It is associated with the root of leguminous plant and help in that is nitrogen fixation. You are familiar with this. Nitrogen fixation is being done, and that is fixing atmospheric nitrogen inside the nodules only. That they cannot do it in free state. These are symbiotic one. Parasitic bacteria, that they used to draw nutrition from the living, that is other livings, and that they have special devices to draw nutrition from livings. The living one is known as host. This living may be animal and that may be plant. Come to the next now, that is reproduction in bacteria. Reproduction in bacteria, it may be asexual or sexual reproduction. True sexual reproduction is that is absent in bacteria. What is by true sexual reproduction? You see here, true sexual reproduction means that is the gametes are formed, there is fusion which make two, that is zygote, and there will be that is meiosis, cell division. All these three, these are absent in bacteria. That's why true sexual reproduction is absent. Come to the next now, asexual reproduction. Binary fission is the most common one, that is takes place by amitosis, and that is mitosis and meiosis. Very important, these are absent in prokaryotes. This type of cell division are found only in that is eukaryotes. Here the binary fission, one bacterial cell, it will start to grow and it will start to divide, that is the constriction will develop, and one bacterial cell that will divide into two. You are seeing this NCERT diagram. Now endospore formation, in unfavorable condition, that this bacteria that develops that is thick wall, and inside this thick wall is resistant one, the content of the bacteria will be protected. In some rod-shaped bacteria, this develops, endospore is formed. This is highly resistant structure, and it is because of calcium 𝕆𝕍𝕍 acid. It will protect the bacteria from unfavorable condition, and when the conditions will be favorable from this bacteria, from this endospore, that is living bacteria will come out. That's why this is source of pollination and dispersal, and it's not source of that is reproduction. Come to the next now, sexual reproduction. We used to call it genetic recombination. Primitive type of sexual reproduction is there, also known as parasexual reproduction. There is no gamete formation, as I told you earlier. There is no fusion. That is no zygote. When the zygote is not formed, so here this is only haploid. 2n situation is never achieved, and there is no alternation of generation. Alternation of generation means from n will get the 2n will develop into n, but we do not have, that is no alternation of generation. Alternation of generation is also absent in bacteria, but the sexual reproduction takes place by, that is genetic recombination. The two bacteria will come in contact, and their genetic combination that will be changed, that will be modified. So this genetic recombination, that may be by three important methods. One is that is transformation, one is that is conjugation, one is transduction. Detail of this you have to study in other chapters, right? Come to the next now, economic importance. Already I have seen chemosynthetic bacteria. These are the autotrophic bacteria, that is oxidize some of the things. For example, nitrite have been oxidized to nitrate. In doing so, that the energy is released, and this energy is used for synthesis of food. And in doing so, they decompose, that is complex or in dead material. That's why they have, that is great role in, that is recycling of nutrients. Soil fertility, very important role. You will be surprised. We used to say that human is the top of evolution, but we are totally depending almost for all the things on other. That is, we cannot have our own oxygen. For this we are depending on plants. That in nitrogen compounds, which are very important for living, that is none of this compound we can synthesize at our own. Soil fertility, you see here, free living, that is they are going to fix nitrogen. Nitrogen, that is there, nitrogen is fixed. Fixation of, that is nitrogen fixation. Free living nitrogen fixing bacteria. This is very important. The four one is Azotobacter, one is Clostridium, one is Klebsiella, one is 𝕆𝕍𝕋𝕼. These are found freely in soil, and they convert nitrogen into, that is nitrogen compounds, ammonia. Symbiotic, as I told you, Rhizobium, found in, that is root, that is nodules of legume. Frankia is found in root nodules of non-leguminous plants like Alnus and Casuarina. They used to live symbiotically and fix nitrogen. Ammonifying bacteria, amino acids are converted into ammonia. You know, if when animal dies, you must have seen a foul smell is there after death of an animal. This is because of ammonia. The proteins what are there in this animal, they will be decomposed with the help of ammonifying bacteria, and important ammonifying bacteria are, one is Bacillus ramosus, one is, that is Bacillus vulgaris. Important one is, now there's a lot of, that is cry for the food, and single cell protein is obtained from bacteria, Methylophilus methylotropus. This is in news. That is, this bacteria can multiply at a very fast rate, can give us a good quality of protein. Vinegar production. Vinegar, you know, is used as preservation of food by Acetobacter aceti. The curd, cheese, this is produced by Lactobacillus. The group is there known as lactic acid bacteria. Then antibiotics, not only obtained from fungus like penicillin, these are obtained from, that is bacteria also. One of the bacteria is, that is Bacillus 𝕍𝕋𝕍. This one very important one. This is Pseudomonas putida. It is known as pollution controller, known as superbug. That I like to tell you a few things about this. What happens in sea when there is an accident and there is oil spill, then that will cause pollution. And how this pollution is to be checked and removed, that this bacteria, Pseudomonas putida, it has been genetically engineering genetically engineering was done, genetically modified it was, and this modification was done by an Indian scientist. This bacteria that will be put here, it will multiply, that is at a very fast rate, and that is it will be able to remove this pollution. Rating of the, that is fibers by Clostridium and that is Pseudomonas, that is these help to remove or to separate the fibers. And disease causing bacteria, you have to remember not many more, only the examples given in NCERT, you have to go for this. That is cholera by Vibrio cholerae, typhoid by Salmonella typhi, tetanus by Clostridium tetani, and citrus canker by that is Xanthomonas citri. Come to the next now. One more group we have to discuss, that is one more type of Monarian, that is Archaebacteria. I told you in the beginning of classification, Archaebacteria have been separated from rest of the Monarians on the basis of some important characters, because they have different type of cell wall and that is they have different type of that is cell membrane. These are the most primitive one, known as oldest living fossil. Archaebacteria, because of the different cell wall or cell membranes, they can survive in extreme conditions, even in that is hostile climate. They can, harsh climate, harsh environment, they can survive easily. Come to the next now. Archaebacteriaceae, types of this. We have three main types of Archaebacteria depending on their habitat. The one is that is methanogens. As the name indicate, at such habitat the methane is produced. That's why methane is produced. They are known as methanogens. These are found in marshy habitat. For example, the rice field is there, or also present in the gut of several ruminants, where they used to digest cellulose and the methane is generated. They are capable of converting carbon dioxide, methanol and formic acid into methane, and because of these are known as that is methanogens. Right? Methanogens, these are called. Now, these are chemoautotrophs. They used to get energy by doing these chemical reactions, and these are obligate anaerobes, means they cannot survive in air or in oxygen. The example is Methanococcus and Methanobacterium. The second type we have, that is halophils. Come on. Halophils, as the name indicate, halophytes, that is salt-loving these are. These are found in highly saline habitat. It may be that is sea water. Yes, it may be that is salty lake. As you know, near Jaipur we have, that is salty lake is there, Sambar. Then it is also found in brine water, where concentration of the sodium chloride is that is very high. These are heterotrophs, not able to make their food, and that is these are that is aerobes. These are example is Halobacterium. We have to deal now, that is thermoacidophils, the third group of Archaebacteria. These are also found in harsh habitat, as the name indicates. It is made up of two words, that is thermo and acidic. Thermoacidophils means these are found at places where temperature is very high, about 80 degrees Celsius, and the medium is acidic one, pH is 2. For example, these are found in hot sulfur springs. These are also chemoautotrophs. They can prepare the food, and these are facultative aerobes. Example is thermoplasma, and that is thermoproteus. One very important point is to be, that is reference is there regarding halophils. You see, halophils, these are having a pigment in their membrane, and that pigment is that is similar to that is the pigment found in human eye, that is bacteriorhodopsin. And with the help of this pigment, that is they can trap the light energy. That is light energy will be trapped, and this light energy will be converted into that is ATP, adenosine triphosphate. But interesting thing here is this ATP is not used in synthesis of food. That is not used in CO2 fixation. You have to remember it. Interesting point this is. That is cyanobacteria, blue-green algae. Initially, This was kept in two kingdom classification. In kingdom plenty, the blue-green algae were kept along with other algae. But because these are prokaryotic one, that's why they have been shifted in, that is, four kingdom classification in kingdom Monera, and even now we used to call them popularly known as blue-green algae, cyanobacteria. Some important characteristic of this: these are the most primitive organism, and they perform oxygenetic photosynthesis. What do you mean by? For the first time, they release oxygen in photosynthesis. They release oxygen in photosynthesis because they use water. Water will be oxidized. As a result of this, oxygen is reduced, oxygen is released. You see, children, that is, this release of oxygen by cyanobacteria that played very important role. That is because of this, that the ozone filter was formed. That is, the earth was covered by this ozone filter. The cosmic rays have been filtered out, and the life has started to come from water to, that is, land surface. That's why BG or cyanobacteria is known as most successful and self-dependent organism. It can do photosynthesis. It can fix nitrogen. That's why it doesn't need anything from any other source. They are mainly freshwater organism, but are found marine also. One important example here is Trichodesma erythraeum. This is unicellular blue-green algae, cyanobacteria, and it causes red sea. It multiplies at such a fast rate that the sea becomes red. They also show symbiotic association, yes, with plants. For example, that is in cycas coralloid root, that is 𝕆𝕋𝔼𝕆 is there. That is, it is having this symbiotic association, 𝕆𝕋𝔼𝕆. That may be unicellular, as I told you. This is one example of unicellular blue-green algae. And unicellular, that is spirulina, which is used as single cell protein. It is rich in protein. It may be colonial, it's like 𝕆𝕆𝕆, or it may be filamentous, that is oscillatoria. This is a diagram of this 𝕆𝕆𝕆 filamentous one. This is colonial one, and that is wrapped in a mucilaginous sheath, which is a protecting one. This is non-motile, and it is having some specific cells known as heterocysts, which are thick-walled, and these are that is responsible for nitrogen fixation. So you see the same thing here. Bacilli are surrounded by mucilaginous sheath. Very important point. Photosynthesis, thylakoid contains chlorophyll B, which is similar to the hair plant, and because of this, they are able to release the oxygen in photosynthesis. The other water-soluble pigments are phycobilies. These are photopigments. BGA can perform biological fixation in anaerobic conditions only. In anaerobic condition, why? Because the enzyme responsible for nitrogen fixation, that is nitrogenase, that needs, that is anaerobic condition. The heterocysts, these are the specialized cell for nitrogen fixation, and that is, these are thick-walled. They do not have, that is, photosystem second. You must know photosystem second, that releases oxygen here. There is photolysis of water, and the oxygen is released. If heterocysts will have photosystem second, then there will be oxygen, and in presence of oxygen, nitrogenase enzyme cannot work. Some of the interesting examples are, that is, nostoc and 𝕆𝕆𝕆. Regarding their reproduction, that is, they reproduce just like other monarians by asexual method. The most common one is binary fission. The fragmentation, that is, one filamentous algae will be cut into pieces by movement of any aquatic animal, and each piece, each fragment is able to develop into new plants. The harmogonia are also formed. How harmogonia develops? That these are the, that is, typical cells. These are some of the cells, they become concave and dead one. And these concave cells, these are, that is, these are known as, that is, these concave cells are the dead one. And because of this, the filament will be divided into parts. These parts, that is known as harmogonia, it develop into the new plants. Come to the next now, that is Economic importance of blue-green algae. They provide fertility. As you have seen, they fix nitrogen. They provide fertility to the soil by nitrogen fixation. They provide food for the animals in that is aquatic ecosystem that they used to do photosynthesis. All aquatic animals that they used to eat it. That's why they provide food. Even for human consumption, spirulina is there, single cell protein. That some of the blue-green algae are used for antibiotic production. One of the example is 𝕆𝕋𝔼𝔼. An important point related to economic importance is known as algal bloom. Algal bloom is excessive, that is growth in water bodies. That this is a water body from the nearby area where nitrogen and phosphorus fertilizer are used. That is excess of this that will leach out. It will come to the water bodies. The fertility of the water body will increase, and this increased soil fertility that will be responsible for excessive growth of this blue-green algae. These blue-green algae, they release some toxic substances in water which may kill the aquatic life, and they also that is consume the dissolved oxygen. Because of deficiency of dissolved oxygen, that is it will cause the death of aquatic animal, and this is known as death factor. It causes death of aquatic animals. Come to the last of this, that is part of lesson, that is mycoplasma. The diagram you are seeing here, some interesting points are, this is the smallest living cell ever known. It is aerobic. It used to prefer live in air and oxygen, but without oxygen also it can survive. Important point that appears in examination many times, that is facultative and aerob. This one is. These are saprophyte, but mainly parasite. This question came. Mycoplasma mainly are parasite, and when parasite, they will cause disease in animal and plants. Cell wall is absent. That's why they can change any shape. These are highly pleomorphic and are known as joker of plant kingdom. And culture of the colony, they appear just like fried egg because the cell wall is absent. That's why this is known as, that is bacteria without, that is coat. The coat, that is the cell wall is absent. These are non-motile. They have both RNA and DNA. DNA is double-stranded thread-like. You are seeing here, that is this DNA that is not confined to the center of the cell. This is dispersed throughout the, that is cytoplasm, extent almost throughout the cell. These are heterotrophs, I told you, and most of these are parasite. They cause disease in plants, which is brome, aster yellow, and little leaf of brinjal are some, that is specific disease, plant diseases. Diseases in animals, 𝕆𝕋 in cattle, why it is known as mycoplasma. Mycoplasma, popularly known as PPLO, 𝕆𝕋 organism, that was discovered by, that is Novak and by Rausk from, that is pleural fluid, which causes pneumonia in cattle and inflammation of the genital organs. Disease in human being, infertility in man, and it causes a typical type of, that is pneumonia. So this is about mycoplasma. So children, that was the part 3, lesson 2, that is biological classification, unit 1, diversity in organism. We have finished it. You know that this chapter will be completed in 7 parts. After completion of all the 7 parts, we have to deal, that is NCERT exemplar questions, and the questions appeared in NEET examination previous years. Thank you very much. The next program will be given to you in slide.